So, this presentation was originally going to be called Coarse-Grained Simulations of Myosin 7 using Fluctuating Finite Element Analysis. Now it's called Will It Bend, to kind of lull people into the trap of thinking that it'll be humorous and light-hearted. In this presentation, I'll be discussing new novel ways to simulate biological molecules, specifically one called Myosin 7, how they work, and what they tell us about Myosin 7 and their broader applications to the wonderful world of biophysics. Myosin 7 is part of the myosin family of molecular motors. It's typically found in the eye and inner ear, and it's involved in the transport of pigment sacs and vacuoles. It's critical to vision and hearing. If you have a mutation in the gene for it, you'll probably be blind, deaf, or dead. This is what it looks like. It has 4 main regions. There's the head and the tail, which have binding sites on them and can bind to various compounds. An alpha helix runs most of the way through the molecule, and this is a cartoon depiction of a helical polypeptide chain. This part of the alpha helix in the middle is called the stable single alpha helix because it has charged amino acids attached to it with ionic bonds between them, which makes it stronger and stiffer than a regular alpha helix. Finally, this bit is called the lever. It has 5 binding sites called IQ domains, and there's a protein called calmodulin bound to each one. This blurry-looking image of Myosin 7 is the absolute cutting-edge picture, the best picture we can get, and I think it's from cryo-EM. A few papers came out a few years ago with the suggestion that Myosin 7, when inhibited, folds in half like this, and that it binds to itself. And it's thought that this behavior is activated by the presence of calcium ions. Unfortunately, this is the clearest picture we have. We don't have a better resolution, and we also don't have any intermediates. Visualizing processes like this can't be done by biologists alone. It requires a cross-disciplinary effort from computer scientists, software engineers, physicists, and coffee. And together they came up with the field of atomistic molecular dynamics, which is a way of simulating biological molecules as big structures made of thousands of connected atoms. As you can see, Myosin 7 is a big, very complex molecule, and it takes a very long time to fold, far longer in fact than atomistic MD simulations are designed to run for. Here's the thing though, the folding that you can see here has nothing to do with what the individual atoms are doing. It has to do with changes in the large-scale structure of the molecule. Given all of this, you might say, well, why do we need atomistic detail at all? Why not just use a model that removes the finer detail but keeps the large-scale structure of the molecule intact, allowing for simulations of biomolecules that are larger and for longer periods of time? Well, if you'd had that idea some years ago, you might have gotten a PhD thesis out of it, but it's too late now. Some Robins already did it, and it's called Fluctuating Finite Element Analysis, or FFEA. All they needed was to work out a way to convert a complex biomolecule into a simple 3D mesh, and then find a system of equations that's able to model it. So this is the basic workflow for turning an atomistic model into a coarse-grained continuum model. The first thing you do is go from your actual atoms to this homogeneous blob called an electron density map, which lets you define the surface by the probability that an electron will be found in a given place. Now, this is why we say continuum, because it's no longer a collection of discrete atoms. It's just a big, continuous blob. We're actually quite fortunate to have atomistic data for Myosin 7 at all. A lot of these bigger motor proteins only have continuum data like cryo-EM data available for them. The electron density map is then converted to a generic 3D model made of polygons, and it's important to note that the EM density map was a volume, whereas this is a surface. The internal structure is removed. There's a special FFEA-specific tool called the Surface Coarse Grainer that reduces the number of triangles in the mesh. The fewer the triangles, the faster it runs, but the less accurate the simulation is. The granularity can be varied, and the more important parts are normally given more triangles. Finally, we convert the mesh from a surface with triangles to a 3D volume filled with tetrahedrons, which is the final model we use in the simulation. Before I show you some results, the math slide. This is the Cauchy momentum equation. It is beautiful, and you should definitely appreciate it. It's really just F equals ma, Newton's second law, for continuum systems. This term here is a viscous drag term that resists any motion in any direction, and this term here is an elastic restoring force, which tries to pull the molecule back into its original shape if you deform it. As part of our simulation parameters, we can actually vary the Young's modulus of the model, and that has an effect on this term. That term, the F there, is just any external force. And this term here, the pi term, is the thermal noise term. This gives every single point, every node, a small random stress every frame. Now, think of this as a consequence of Brownian motion, and think in particular of the fluctuation dissipation theorem. At small scales, dissipative processes can work in reverse. Normally, you lose kinetic energy to friction or drag because it's converted into heat. Here, it's the heat that's supplying the energy needed for the molecule to change shape. So this equation, which has a fluctuating thermal noise term, is solved by a number of numerical methods, including finite element analysis, hence the name fluctuating finite element analysis. So here's an example of what an FFEA simulation of myosin 7 looks like. This is pretty much the first thing I did. I parameterized it with dummy values. In reality, myosin 7 doesn't have moves that are anywhere near this sick, but it does show approximately how it moves and how it has to bend for self-binding to occur. So just for reference, again, this is what it ought to look like, and you can see in the video that it got pretty close to this conformation. So this thing, on the other hand, is a simulation of myosin 7 that has been parameterized using stiffness data from some papers written about myosin 6, which is roughly the same. So this is parameterized using experimental stiffness data. And as you can see, unfortunately, this one is doomed to wander around forever without ever self-binding. So you might have come to the conclusion that this is actually a sort of null result, but there's actually one more thing we need, one more ingredient to make this self-binding work, and it's these things, the calmodulins. Now, calmodulin is weird sort of biology jargon for calcium-modulated protein. According to another recent paper, in the presence of calcium ions or a chelating agent, some of these calmodulins can unbind from the IQ domains. This leaves behind an unstable alpha helix. The ionic bonds around the outside of the stable alpha helix, you remember on the other side, protect the inner hydrogen bonds from being attacked by water molecules. But the alpha helix on the right-hand side doesn't have those. As a result, it behaves kind of like the cord for a pair of earphones. It immediately tangles itself into an unusable mess. So rather than being a long, thin structure, it becomes a short, folded structure. With that in mind, these are the variants that I used of the model. In each case, I ran a simulation on the collapsed and uncollapsed versions of myosin 7 in the presence of calcium ions and the chelating agent ETGA. As you can see, they cause different calmodulins to unbind. Now, we don't know how the structure of the alpha helix will end up, but because that detail is going to be coarse-grained away, it's not really important. We can just ignore it. So this graph shows the end-to-end distance, that is how close the binding sites are on the y-axis, against the active area Young's modulus. So simulations on the right-hand side are stiffer, and simulations on the left-hand side are more flexible. And the orange line is an estimate of the maximum distance at which binding can feasibly occur. So the simulations in this corner here are all simulations where the inhibitors are present, and these ones over here are the ones where the inhibitors aren't present. So that's great. You might think, that's exactly the result you would expect from the experimental data. And yes, okay, it is nice. We have a FFEA model of myosin 7 that agrees with the results of experiments from multiple papers, very different to our null result. But even though this is nice, there's still a hell of a lot to do on myosin 7 in FFEA. For example, you may be wondering, can a coarse-grained simulation ever reliably model a second-order structure like an alpha helix, especially a collapsed one? And the answer is probably to investigate using MD simulations to model the collapsing alpha helix and see whether it ends up in a state that can be represented using FFEA. Similarly, we need to look at better ways of setting the material parameters of our models. So right now they're dependent on a number of assumptions and approximations, but there are methods being developed right now for getting more accurate values for stiffness using experiments involving quantum dots. You basically attach the quantum dot onto the end of the molecule, and then you get the stiffness from the Gaussian distribution of its position. And of course, the obvious next step is to run more simulations for longer and to add in the things that we're missing, like the binding site potentials. So this is dynein, another motor protein, and it's taken years just to get it sort of walking along an actin filament like it's supposed to. So for myosin 7, this is really just the very beginning of a long and arduous journey. Molecular modeling has kind of a reputation for only being useful in a small subset of biological systems, but FFEA is aimed at simulating larger and more complex systems that atomistic molecular dynamics can't or shouldn't be used to model. And when you get to mesoscale processes like the formation of blood clots out of fibrin, atomistic molecular dynamics stops being an option altogether. In other words, there's a gap in our current simulation knowledge. We can model everything on the left-hand side and everything on the right-hand side, but we're missing this information sort of right in the middle, and this region is where some of the most crucial biological processes lie. FFEA is a technique that has the potential to greatly expand the scope of our molecular modeling capabilities, to bridge the gap between the nanoscale and the mesoscale, and to probe into biological systems and processes that have so far been beyond our understanding. And to me, that's very exciting indeed. Sorry about this slide. I forgot I had that in there. So, yeah, that's the end. Here are my sources. Thank you very much for listening.